All right, welcome to part 3 of chapter 3, where we're going to look at the structure of eukaryotic cells. Now, eukaryotic cells are found in all organisms of domain Eukarya. So this is kingdom Animalia, which would include us, kingdom Plantae, which are all the plants, kingdom Fungi, the fungi, and then kingdom Protista, which are all the eukaryotic organisms that don't fit in any of the other 3 kingdoms. So this would include protozoa, this would include algae, slime molds, water molds, lichens, and a few other organisms. The figures that you see are pretty typical to what you would find in an introductory biology textbook, where they're showing an animal cell up top and a plant cell down below. And there's a lot of things that they share. I mean, if you're looking through the lists, you see that there are a lot of structures that are common to all eukaryotic cells. But there are some specialized structures the plant cell has that the animal cell doesn't have, like this large central vacuole, which holds water for photosynthesis and increases the pressure inside the cell. They have chloroplasts, which are the green-shaped or green-colored organelles, which are for photosynthesis. And they also have a rigid cell wall. If you're going to store excess water inside, that's going to increase pressure, and the cell wall keeps you from bursting. So lots of common structures in all eukaryotic cells, but there are some specialized structures that plant cells have that animal cells don't have. And really we should look at a fungal cell too, because fungi have a cell wall that's rigid, much like a plant cell does. When we go inside of eukaryotic cells, one of the first things we notice is there's a whole bunch of membrane-bound organelles that we didn't see in prokaryotic cells. And remember, I said a lot of biochemical activity is associated with membranes. This is because over time, and remember, biological time is 3.5 billion years, membranes, the proteins that a particular membrane has within the membrane have specialized. So, for example, the proteins in the nuclear membrane may be very different from proteins in the rough and smooth endoplasmic reticulum or the Golgi apparatus, etc. And this membrane system, endomembrane system, has kind of evolved to work together to allow the cell to take information that's stored in the nucleus, use it, rewrite it so it can be understood by proteins in the rough and smooth endoplasmic reticulum to build product that then gets passed on to the Golgi apparatus to be further refined or put together to form more complex structures or possibly be stored for a while, and then use that to either add membrane to the plasma membrane of the cell so it can get bigger, or to produce specialized organelles like lysosomes and peroxisomes, or create temporary storage vesicles that we call vacuoles. And so, altogether, this is how the cell is able to specialize in making all the various things that the cell needs. We'll start with the nucleus. The primary function of the nucleus, don't tell me it's the brain of the cell. Their cells don't have a brain. The nucleus can't think by itself. What it does is it holds the chromosomes. And if there are any nucleoli, they're going to be there too. Chromosomes have your genes. And, you've got, on average, 46 chromosomes in humans. There's a few of us in the planet that have a few more, or maybe one more or one less. But the vast majority of us have 23 pairs. And we got a copy of each pair from our biological mother and a copy from our biological father. And on our chromosomes are our genes. And genes code for proteins that either have a function, that either build other things, or that regulate development or how we respond or something to that, some function of the cell. The nucleoli help to make ribosomes, and it's ribosomes that build the protein. So this is the big part of what the nucleus does is it stores the chromosomes, keeps them secret, keeps them safe. If I was Gandalf, that's what I would say. Nucleus has a nuclear envelope that's a double membrane, and it has pores in that open directly into the endoplasmic reticulum. And they're heavily gated, which means that the nucleus very tightly controls what can get into or out of the nucleus. And again, it's bound to the endoplasmic reticulum. And that makes sense. If the information for how to build something is on the gene, when we rewrite that into a message, we want it to go directly into the endoplasmic reticulum where we can use it to build things. And here you see just kind of a diagram showing, a nuclear membrane with a double membrane and then pores that open directly into the endoplasmic reticulum. This endoplasmic reticulum is rough with ribosomes, so this is where we're going to build proteins. You can see the chromatin that makes up the chromosomes that are unwound. Here's a big old nucleoli. Okay, and again, this is just a diagram, but it gives you an idea of the various things that are present. Endoplasmic reticulum endo means within. Plasma refers to the cytoplasm. And when something reticulates, it winds back and forth. And so what this really is a whole bunch of sacs and tubes that have membrane that are attached to the nuclear envelope and wind around it, back and forth around it. And with all that extra membrane, we have a lot of specialized function. If the ER, the endoplasmic reticulum, has ribosomes attached to it, then we call it the rough ER. And ribosomes build proteins, so that part of the ER is going to make proteins. The smooth ER lacks ribosomes, and so this is where the cell is going to make complex lipids like the phospholipids of the cell membrane and carbohydrates that it's going to need to, make all the various membrane or all the other structures that the cell has. The Golgi apparatus is going to be, again, more specialized membrane. There are a bunch of flattened discs that are side by side. There's a side that's closest to the endoplasmic reticulum called the cis side. The farthest side away is called the trans side. And what happens is materials that are built in the rough and smooth ER are then packaged into pieces of the ER that pinch off as a vesicle. So you got whatever was made inside of membrane from the ER that then drifts across, and the membrane fuses to the first cistern, which is what the discs are called, of the Golgi, and empties its contents to the inside. Now, sometimes the Golgi will just store things for a while. Other times, it'll put things together, like it might add phospholipids together with proteins. It might add two polypeptide sequences, primary structures of a protein together to build a secondary or a tertiary structure or a quaternary structure. But the Golgi is sorting and packaging. And then the things that it needs to send either out into the cell or out of the cell are going to be packaged in membrane from the Golgi to form a transport vesicle. See, material very rarely just moves through the cytoplasm when it's not surrounded by membrane. And so it's a big job of the Golgi to package these materials for transport. Here you can see the Golgi apparatus. It's got these flattened discs called cisterns. The side that's cis is closest to the ER is over here, and this trans face is the farthest away. It's on the trans side that anything the Golgi is putting together in a different order than it got from the ER. That's where they're the most advanced or most finished. Some of the cisterns are storing material inside until it's needed to make more product for the cell. Again, vesicles are going to be membrane-enclosed sac-like organelles where we just pinch off parts of the Golgi apparatus. Now, we might make a specialized organelle like a lysosome or peroxisome, where we're going to take, in the case of a lysosome, we're going to take proteins that are digestive enzymes. We're going to attach them to a chaperone protein that keeps them from being active, and then we pack them in a lysosome. And then we release the lysosome out into the cytoplasm. And we'll see in a minute how the lysosome is involved in digestion. Peroxisomes break down toxic substances and free radicals in the cell. So they're kind of like a lysosome, except they're very specific about what they target. And then vacuoles are kind of like the Tupperware of the cell. They're for temporary storage or transport. Here we see a macrophage, which would be one of your white blood cells of your immune system. And here this macrophage has found a bacterium that doesn't belong. So it phagocytizes it, wraps itself around it, and takes some of its cell membrane and creates a vesicle with a bacteria inside. And then that vesicle fuses to a lysosome. Once they fuse, those enzymes, those digestive enzymes in the lysosome are activated, and they break down this bacterium. And then whatever nutrients the cell can get from that, it'll absorb through the membrane of the vesicle. And then the rest of it is waste, which can migrate to the edge of the cell and release the waste outside of the cell. Okay, so this is the way that lysosomes are involved in digestion. And it doesn't have to just be, something that has been ingested by the cell. This is how we get rid of extra organelles or used organelles or damaged organelles. We take and package them in a vacuole, and then we fuse it to a lysosome, and we break them down and recycle them. There are also a couple organelles in eukaryotic cells that are very much like bacteria or prokaryotic cells. These are the mitochondria and chloroplasts. And there's a third one that's just recently been identified that I think it's called a nitroplast. But this is really new. This is within the last 6 months that they found that. And in 6 months from summer of 2025. So if you're listening to this summer of 2026 or later, it was in spring of 2025 that they identified the nitroplast. The job of the mitochondria, don't tell me they're the powerhouse of the cell. That's a 4th grade definition. Mitochondria produce most of the ATP in cells that have mitochondria. But realize there are some eukaryotic cells that don't have mitochondria. And there's ATP that's produced in the cytoplasm. They don't make all the ATP. Chloroplasts, their primary job is to build sugars during photosynthesis. And we'll examine the role of mitochondria and chloroplasts in a couple of weeks. Right now, it's enough just to know their structure. Know that they both have their own DNA. These are organelles that they have their own DNA. They have their own ribosomes, and the ribosomes are like bacteria ribosomes, not like eukaryotic ribosomes. They can reproduce by themselves when the cell needs either more sugars or more ATP. They all have double membranes, like they were ingested and put into a vacuole, but not digested. They use waste products from the cell and produce high energy products that they share back with the cell. And when they reproduce by themselves, it's by binary fission, the way bacteria reproduce. So what all this means is that scientists very much believe that at one time mitochondria and chloroplasts were free-living bacteria that got ingested by a larger cell but not completely digested right away. And that over time, and remember, time is billions of years, over time, they became organelles within the bigger cell. And it's to the point now where cells with mitochondria or chloroplasts cannot survive without them, and the mitochondria and chloroplasts can't be grown in the lab outside of the host cell. It's an obligate relationship. And so when one thing lives inside the other, that's called endosymbiosis. Symbiosis is living together. Endo means one inside the other. And there are lots of examples in biology of organisms living together, one inside the other. For example, the trillions of bacteria living in your gastrointestinal tract. They're living endosymbiotically with you. Okay? And when the relationship becomes obligate, then they have to live this way. And there are lots of examples of that in biology. Here's showing you an electron micrograph of mitochondria. You can barely make out the inner membrane with an outer membrane. Okay, and we'll look at the structure and where in the mitochondria various things happen in a later chapter. If we look at a chloroplast, we see, okay, we got thylakoid membrane in stacks called granum, double membrane. Look at all the membrane in a chloroplast. And remember, more membrane, more biochemical activity. This is what makes chloroplasts so efficient. And chloroplast is misspelled up there. This is what makes them so efficient in producing sugars. The cytoskeleton is a whole network of protein filaments on the inside of the cell. And some of them are microtubules, some are microfilaments, some are intermediate filaments. In Bio 100, it's not so important that we distinguish too much between these. What's important is we understand that collectively they crisscross through the inside of the cell to help support the cell. It keeps the cell from falling in on itself. You know, if you take a water balloon, you fill a water balloon, you set it on a tabletop, it's going to swoosh down and flatten out. Well, we don't want cells to do that. We want them to be able to keep their opposite sides apart. This is one of the jobs of the cytoskeleton. Another is to organize things within the cytoplasm. So we don't have all the organelles of one type on one side and all the others on the other. We allow different organelles to be spaced out within the cytoplasm so that wherever we are inside the cytoplasm, we can control all those biochemical activity that's going on so the cell can function at its optimum efficiency. If they're attached to the outside, it allows the cells to move the eukaryotic cells. So these are going to be cytoskeleton is going to be connected to exterior organelles like cilia and flagella to allow the whole cell to move. The cytoskeleton also will work with special proteins called motor proteins that will, let's say, one end of the motor protein will grab onto a lysosome and the other side will grab onto microtubules of the cytoskeleton and will physically move along the microtubule to move that lysosome from one part of the cell to another. And these are really cool. You can actually see this in, I think there's a website coming up. You'll be able to go to that and see this happen. So this is showing you intermediate filaments, microfilaments, and microtubules that all work together to build the cell's cytoskeleton. Motor proteins, again, here's that website. Go check this out. You can watch this and see how they depict this movement of a motor protein carrying something from one part of the cell to the other. Okay, it's really kind of cool. On the outside of the cell are for organelles of locomotion. We typically think of flagella and cilia. They have very similar structure, except flagella are less numerous. They're long and they're whip-like. The cilia are shorter, more numerous, and they tend to move in a synchronized fashion. And so organisms with flagella on the outside, they move, but it's kind of jerky movement, whereas organisms with cilia are really fast and speedy. And we'll see this when you're looking at your pond water cells down the road. You're going to see protozoans inside your pond water samples, some with flagella, some with cilia, and you'll see how they move differently. Sometimes the cilia and flagella are to move the whole cell. Other times they create feeding currents. So they can cause the water to move in a particular fashion so that it draws nutrients to a certain part of the cell. Many protozoans do this because they have an organelle in the cell called a cytosome, which it kind of acts like a gullet to bring food into the cell. Both of these project away from the plasma membrane, and microtubules are going to allow for these to function. Examples of these in your body: you have ciliated cells along your airways that are constantly sweeping mucus and whatever got caught in your mucus up and out of your airways so your lungs don't fill up with dirt. Flagella is what allows sperm cells to swim. There are also pseudopods that eukaryotic cells can have. Pseudopod means false foot. And what this is a temporary protrusion where the eukaryotic cell, and here you can see it in this cell is just kind of reaching out in this direction. And this forms the false foot of the pseudopod. Now, there could be something out here that released a chemical like CO2 that would tell this cell, hey, there might be something out here that we can eat. And what they'll do when they get there is they'll wrap around it with their pseudopods and then pinch together and create a vesicle with whatever was outside inside, and then this drifts into the cytoplasm, binds to a lysosome, and we can digest it. Amoebas behave like this in the ecosystem, the environment, but you also have white blood cells that are part of your immune system that behave and function and move very much the same. This would be your neutrophils, your macrophages. On the eukaryotic cell surface, there are also specializations. Now we saw in prokaryotes that there might be a capsule. Eukaryotic cells may have what's called an extracellular matrix. Okay, this is usually a complex mixture of substances. It isn't just carbohydrates like we saw in the prokaryotic cells. This helps to keep the cell. This helps to keep the cell intact to a point, especially if it's a cell without a plant cell wall, like an animal cell. It also allows animal cells to bind together to form tissues. And this also functions in cell signaling. There are molecules that are embedded in the extracellular matrix that are also connected to the cell membrane, and it allows them to bind to a molecule outside the cell, which then triggers a cascade of changes which sends a message to the inside of the cell. So this is how cells can respond to changes in the environment. Now, cuticles are a special type of extracellular matrix that are found in plants and some arthropods, where the cuticle is waxy and it's waterproof. If you've ever looked at the top of a leaf, you'll notice it's shiny, or fruit, it's shiny. That's because of the waxy cuticle that's trying to keep moisture in and not letting it escape. And then there are other eukaryotic cells that actually have a cell wall, like plant cells, fungal cells, algal cells, where the cell wall provides a tremendous amount of support, gives a very definite shape to the cell, and it's hard to get through the cell wall, so it protects the cell from viruses and bacteria that might otherwise infect those cells that have a cell wall. Here's an animal cell glycocalyx. So we see the phospholipid bilayer, we see proteins that are embedded. Here's our cytoskeleton on the inside, and here's our extracellular matrix, which has fibronectin connecting collagen fibers. We also see that there are proteoglycan complexes with proteins and carbohydrates all connected to the polysaccharide. You put this whole thing together, this is the glycocalyx. All your cells have that. Plant cell walls, this is a cross-section of a leaf. You can see the waxy cuticle on the surface. That would be shiny, and that helps to keep water from just diffusing out of the leaf. The photosynthetic layer is in this area. We'll do a photosynthesis lab later in the semester where we'll talk about the different regions of the leaf and how they're involved in photosynthesis. If we want to hold animal cells together, we're going to do it with what are called junctions. And these physically connect one cell to another cell. And there are three basic types of animal cell junctions. A tight junction is waterproof. Okay, it firmly joins epithelial cells together to keep any fluids from leaking through them. Okay, but there are also adhering junctions, also called desmosomes, which anchor cells to each other but not as tightly. And so water can pass through between these. And then finally, there are what are called gap junctions, which is kind of like a portal from one cell to another. So let's suppose you had your kitchen was next to your living room or your family room, and you had a hole in the wall so people could pass something from the kitchen, snacks from the kitchen right into the family room, or the empty plates and platters back into the kitchen. There's a hole in the wall. Well, that's kind of how a gap junction is. It's a hole between cells that join, so cytoplasm and other materials can pass freely from one cell to another. Plant cells are already tightly anchored to each other because they have a rigid cell wall. And when plants develop, their rigid cell walls are anchored tightly to each other. So they don't need tight junctions or desmosomes. But they still need to pass materials from one cell to the next. And so the holes that do this are called plasmodesmata. And this lets water, nutrients, signaling molecules, waste materials to flow through from one cell to the other. Plasmodesmata, one of my favorite biology terms. If I ever had a kid, I'd name him Plasmodesmata. I'd call him Plasmo. All right, that's why I'm not allowed to have children. Anyway, here's the different junctions. You can see the plasmodesmata, the gap between two plant cells, their cell walls firmly anchoring them to each other. Here you see tight junctions that are so tight that water can't even get through. Here you see the desmosomes, which anchor cells, but interstitial fluids can still pass around the cells, which is important for allowing gases and nutrients to get to each of these cells. And then finally, you see the gap junctions that can form between animal cells to let material freely flow in between. So that's the end of this part of Chapter 3. When we get together next time, the next week, we're going to, we got a little bit to cover in the rest of Chapter 3 in terms of membrane transport. And then we've got some material that'll be covered in Chapter 4 on energy and enzymes. And then the following week, we'll finish up Chapter 4 and get started on 5 and then finish with 6. So we skip around a little bit over the next couple weeks. But next week, we'll start off learning about energy and the first 2 laws of thermodynamics.